Thank you for the introduction. It is my absolute honor to speak here at MHIS 2023. I'm a researcher at the Precision, Personalized, and Translational Neuroscience Lab here at Stanford Med. So today I'll center my talk around precision therapeutics and sharing our lab and my personal work. Existing diagnostic and treatment systems in psychiatry are not optimized. Patients with depression or anxiety disorders are usually first put through a first-line antidepressant, and according to research, as much as a third of patients do not respond to this first-line treatment. Then what do we put them through? So first, if they fail to respond, we give them a different medication based on clinicians' experience, and then we go through this trial and error process until we find a medication that either helps the patient reduce their symptoms or they hit remission. So the self-reports, which is given at the beginning before treatment, unfortunately does not give information on how we should treat the patients. It also places patients into a highly heterogeneous bucket, which is depression. The problem is that we risk conflating people who demands different treatment into this very large bin. So our lab proposes an alternative using neuroimaging, which uses a data-driven approach to achieve a more homogeneous grouping of these participants based on their brain biology. We will make more informed treatment decisions based on these groupings, and ideally, some group responds better to certain medications, which would inform personalized treatment. So how exactly do we do this grouping? I like to make an analogy to cardiology. In the 1950s, imaging played a huge part in breakthroughs in cardiology, which ended up halving the number of patients who die annually from heart disease. Today, thanks to the advances in human functional neuroimaging, we now have the foundations for deploying imaging metrics that can quantify brain circuits. So our lab focuses on 6 tractable circuits derived from fMRI because they are well implicated in depression as well as in the mechanisms of treatment. The 3 circuits on the left are likely already familiar to you. They are default mode, the salient circuit or network, the frontal parietal network, also known as frontal executive, and notably, all of these 3 circuits are imaged under task-free conditions. On the right, we have, in task-evoked conditions, negative affect involving the amygdala and limbic connections probed by a negative stimuli, positive affect or reward probed by a positive stimuli, and cognitive control probed by effortful inhibition tasks. So how then do we measure these circuits at an individual subject level? Over the past few years, our lab has developed a rigorous quantification platform for fMRI. And the first role is how we go about defining the circuits in a standard way in any given patient. And in row 2, we then extract these anatomical features to compute numerical metrics for any given circuits. So at this step, we are quantifying the strength of connection at rest between the regions or the physiological interaction with analyses and tasks. And then we are expressing these metrics relatively to the healthy reference groups in units of standard deviations. If you're interested at all in how we derived these circuits and the psychometric properties of these circuit biotypes, I encourage you to read our paper, which is cited on this slide, for more methodological details. To further validate our theory, we studied how these circuit scores map to discrete symptoms and behaviors in anxiety and depression. So here we have constructed radioconnectivity plots that maps different regions of the brain networks to symptoms. For example, you can see in the salience networks, which is the second network here, particularly the connection from the insular to amygdala maps to specific symptoms of anxious avoidance, anxious states, and this usually connects to patient reports of having their heart racing, having hot and cold sweats. So now that we have established that these individualized and standardized brain circuits are reliable and robust, how do we make them clinically actionable? To do so, we need to solve one big problem, which is converting the continuous circuit scores into discrete categories for clinical decision-making. I optimized the threshold for circuit dysfunction in terms of standard deviation from healthy controls. And based on analyses, 0.5 was chosen as the threshold to maximize the sensitivity and specificity for predicting treatment status when we use brain circuits as inputs. So now that we have established the circuit biotypes and threshold for clinical dysfunction, we applied the system to a new data set. This data set, called the iSpot-D, contains the psychosocial metrics and neuroimaging of about 200 patients, as well as more than 130 healthy controls. Notably, 58% are female, compared to much higher proportions of female participants in the same type of studies. So participants were randomly assigned to either SSRIs or SNRIs, which are commonly prescribed antidepressants, over the 8 weeks of treatments. And then at the beginning of end of the 8 weeks, we get them self-reports, measure their symptom improvements, and we also have them go through multiple fMRI scanning sessions at the beginning to assess the networks that I've mentioned above. With this powerful data set, we could study how the circuits distribute within this sample. I found that 97% of patients with depression have at least one circuit dysfunction. This is encouraging in showing that mental health disorders are indeed quantifiable in the brain, not just on questionnaires. And to make this dysfunction clinically meaningful, we broke down this 97% into smaller buckets to identify a subgroup of patients, which connects back to the first slide of the rationale that we want to break things down and give them personalized treatment. So to talk about this smaller buckets, we first need to understand a concept which we define as primary circuit dysfunction. If we look across all the individual subjects, we can assign them their primary or their most extreme dysfunction in brain circuits at a minimum of 0.5 SD away from healthy reference. In here, that you can see all the circuits are represented in the iSpot-D dataset. So this is a plot of their primary circuit dysfunction. Although all the circuits are represented, there's significant variation in the distribution of each dysfunction. The latest work in our lab involves predicting treatment outcomes from the circuit biotypes. So after parsing the brain data of each participant into the 8 brain circuits of interest, I determined circuit dysfunction using the optimized threshold, and then entered the circuit dysfunction information into a cross-validated regression model trained on multiple data sets, which predicts remission and response to treatment in any given participant. In this figure, we undertook a model comparison, contrasting the blue curve, which is the brain circuit-based curve, to the red curve, which is our null model. So in the blue curve here, the input of the model is the scores of all of the circuits I've mentioned above. And for the red, it is a null model that is trained purely based on the self-report at baseline. What we found is that the circuit model achieved an 81% accuracy compared to the null model, which has predictive power only around a random classifier. So this demonstrates the potential for developing better treatment plans based on brain data to improve upon the current treatment paradigm. In the last few moments I have, I'd like to share our translational therapeutics approach to the clinic. So our center has affiliated clinical team in VA Palo Alto, as well as a precision clinic. We are currently working on a digital report system. It has structural information in the middle panel, the circuit scores on the left panel, which breaks down into the circuits we talked about and showing how dysfunctional each circuit is with the patient. And the right panel shows symptom severity, or the self-report scores. The ideal is to use this platform to communicate data to participants in the study, and they are very interested in it. So participants tell us that they really value the tangible feedback on how their brain is functioning, and seeing the biology behind their mental health illness helps to deal with stigma, and they engage more with treatment. I just want to end on a final note that mental health disorders are quantifiable in the brain, and insights from neuroimaging could lead to better and more effective treatment. Thank you, and I invite your questions.
Is phenomenal and
Thank you.
appreciated, because I practice medicine, and the disappointing outcomes from antidepressants are frustrating, and this gives me hope. So I appreciate you.
Thank you so much.
Great. Awesome. Thank you so much, Evelyn.
Thank you.